As we have seen earlier that the statistical approach to find the genes in the genome rely on the statistical variation that is present in the exon and the introns regions. On the other hand, it could be a very straightforward approach, and in this approach we can find the open reading frame. And we know that the open reading frame comprises of the region that is transcribed into mRNA and then protein. If we assume that the length of the genome is n and we get the different sequences of the genome within the genome by dividing divide n with the 3, because we know that there are 3 stop codons present in the sequences of the genes, and these stop codons are TAA, TAG and TGA. And whenever there is a stop codon, the gene is separated and then the intron will start. So we can divide n by 3 and we will get the subsequent the sequence and the short fragments of the within the genome. And we know that the subfragments of these start from the start codon, that is ATG, and that is present in just in front of the open reading frame ORF. And we also know that the open reading frame, it overlaps, because we know that whenever there is a transcription, then after transcription and then the process of translation, then there are 6 frames that are generated in the process of transcription and translation. And we also know that these 6 frames, 3 start from the right side and 3 start from the left side. And these 6 frames are generated because when there is a process of transcription, one nucleotide that starts from the start of the gene of the open reading frame, that is deleted. And in the second frame, the first 2 nucleotides are deleted. And in the third frame, the first codon is totally, whole codon is deleted. And this process also takes place from the other side of the gene. And in this way, the 6 reading frames, they are obtained. So we can see here the 6 reading frames. So the reading frames that are generated from the left side, they are 1, 2, and 3. And the frames that are generated from the right side, they are also 3, 1, 2, and 3. And we can see that whenever there is a deletion of one nucleotide, the stop codon will generate it, or they are generated in different locations within the proteins. So we can see here in the first sequence of the protein, this stop codon is here. Here the translation process will stop. Again in the second frame, we can see that the stop codon is generated within the full length of the protein sequence. And this third one, that is the correct one, because there is no stop codon within this sequence. Similarly, the 3 frames will be generated from the right side. So total will be 6 frames. And the largest open reading frame will be selected as the gene that will code for the protein. One would expect to find frequent stop codons in non-coding DNA, since the average number of codons between 2 consecutive stop codons in random DNA should be 64 by 3, and that comes out to be 21. This is much smaller than the number of codons in an average protein, which is roughly 300. Therefore, open reading frame longer than some threshold length indicate potential genes. However, gene prediction algorithms based on selecting significantly long open frames may fail to detect short genes or genes with short exons. So the problem is by using the statistical approach, we have to consider the short open reading frame instead of the long reading frames. Many statistical gene prediction algorithms rely on the statistical features of the protein's coding regions, that is the codon usage. So by using the codon usage, we can enter the frequency of occurrence of that each codon within a given sequence into a 64 element codon usage array. Here we can see a chart that is the codon usage array, and the genetic code and codon usage in Homo sapiens means humans. So the codon for methionine or AUG also acts as a start codon. Here we can see this is AUG and methionine, because the methionine is always coded with this codon AUG. So it will be 100% because there is no other possibility to code the methionine. We know that all proteins, they start with generally the codon that is AUG, and it is also called the start codon. The numbers next to the each codon reflect the frequency of that codon's occurrence while coding for the amino acid. So we can see that these are the numbers in front of each amino acid, and it shows the frequency of that codon. For example, among all the lysine residues in all the proteins in a genome, the codon AAG, we can see here AAG codon, it codes for lysine, and whenever this codon is used to produce lysine, its frequency is 25%. Similarly, the codon that is AAA, and it also codes for lysine, and its frequency is 75%. So we can see here that the same amino acid is being produced by two different codons, but the frequency of each codon is very much different from each other. So similarly, we can see that, for example, we can see here in this block, there are 4 different codons, and all 4 codons, they code to the amino acid proline. But we can see that the codon CCG has the highest frequency value of 51. And the other interesting thing is that when we sum up all of these frequencies, it will come up to the approximately 100. So we can see that for leucine, this CUG has the maximum frequency as compared to the others, and so on.